My name is Jose Torres, and my presentation is on myostatin-related muscle hypertrophy. Introduction. Myostatin-related muscle hypertrophy is a monogenetic disorder which is caused by a mutation in the MSTN gene located on the arm of chromosome 2 in position 32.2. Myostatin-related muscle hypertrophy is inherited through an incomplete autosomal dominance. Incomplete dominance would be when the phenotype of the heterozygous genotype is distinct from and often intermediate to the phenotype of the heterozygous genotypes. An example would be snapdragon flower. Its color is homozygous for either red or white. The MSTN gene's primary function is regulation of muscle growth, but individuals with the mutation will have unregulated muscle growth and low body fat. The normal gene and protein. Myostatin-related muscle hypertrophy is caused by an incomplete autosomal dominance in the MSN gene. The gene is responsible for regulation of muscle growth and is a part of transformation growth factor 8. The normal gene and protein, part 2. The gene is located on, as I stated before, chromosome 2. In muscle cells, myostatin exists as promyostin, uncleaved, and is not cleaved until the myostatin is in the extracellular matrix. The gene to protein is broken down in detail in figure 1. This image below, it just shows you its position of the gene. Figure 1 gene. The MSTN gene is 700. The MSTN gene has 7,030 base pairs. There are a total of 375 amino acids, which this gene is composed of, and it has a molecular mass of 42,750 daltons. The normal gene and protein, part 3. The MSTN gene is a transformation growth factor beta family member, and in skeletal muscle, it inhibits the stimulation and distinction of satellite and resident stem cells. The metabolic pathway which is involved in the final gene product is the secretory pathway in the Golgi complex. The normal gene and protein, part 4. The process begins in the cytosol, and then to the rough endoplasmic reticulum, and then using a ER to Golgi transport vesicle, the protein moves Golgi cristae from the secretory vesicles that move the protein to the cell surface. The genetic change. Myostatin-related muscle hypertrophy has an unknown frequency, making it quite rare. There is no method for tracking this condition. Individuals with a mutation in this gene show an increased muscle mass and decrease in body fat. And there is no known complications which occur from this mutation. That sounds pretty nice. Wouldn't anyone like to have increased muscle mass and lower body fat? I guess not anyone, but, the inheritance pattern. Being an incomplete autosomal dominant monogenic disorder, in order for a child to be born with a mutation in the MSTN gene, a parent has to be heterozygous and will have increased muscle mass and or a variant of the MSTN gene already. The inheritance pattern, part 2. Each child of the parent will have a 50% chance of inheriting the MSTN gene. And there's a 25% chance that the siblings of a child with the monogenetic disorder in the MSTN gene will also have myostatin-related muscle hypertrophy. There is a 50% chance that a causative variant of the gene will show up in the siblings, and a 25% chance of not having the monogenetic disorder. These are just some references which I used for my presentation.